Welcome back to the deep dive. Today we're tackling something really fascinating in genetics, looking at diseases that, well, they just don't play by the usual rules of inheritance.
That's right. We're diving into a peculiar phenomenon where our own DNA seems to almost stutter on a molecular level.
Just stuttering gene.
Yeah,
Okay, so the topic is specifically diseases caused by trinucleotide-repeat mutations.
exactly. And they're incredibly important, especially for understanding some really devastating progressive neurodegenerative conditions.
Right. So our mission today is to really unpack this unique class of single gene disorders. We want to understand how just a small, unstable bit of DNA, essentially a repeating three-letter sequence, can, expand and cause such complex and severe diseases. We're talking things like Fragile X syndrome, Huntington disease.
hmm And the fundamental idea, the place to start, is defining the mutation itself. It's the expansion of a stretch of three nucleotides, repeating over and over again.
Trinucleotides.
Yes. Usually they involve the nucleotides guanine and cytosine G and C. When this repeating stretch expands beyond a certain number, a threshold, it impairs the gene's function. The DNA sequence itself is inherently unstable.
You mentioned GNC. Is there a reason those particular bases are often involved? It feels like a molecular clue.
Oh, it absolutely is. G and C bases actually form three hydrogen bonds between them. A and T, adenine and thymine, only form two.
So GC pairs are stronger.
They are, which makes the DNA double helix very stable in that region normally. But, and here's the paradox, that strength makes the single strand, when the DNA separates during replication or repair, highly prone to folding back on itself.
Folding back.
Yeah, it forms these things called secondary structures. Think hairpin loops or even more complex shapes like train plexes because of those strong GC bonds holding the fold together.
So the DNA literally ties itself in knots, almost.
In a way, yes.
Okay.
And then, when the enzyme that copies DNA, the polymerase, encounters one of these knots, these secondary structures, it can get confused. It might pause, slip, or stall.
Okay.
And when it restarts, it sometimes recopies the section it just made. This slip-up leads to an expansion of that repeat sequence in the new DNA strand. That basic instability is the root cause. #I. General principles and pathogenic mechanisms.
Okay, so this whole mechanism, this expansion, it's actually a relatively recent discovery in the grand scheme of genetics, isn't it?
Oh, absolutely. It really turned modern genetics on its head. For years, maybe even centuries, people observed strange inheritance patterns in diseases like Huntington's or myotonic dystrophy.
They saw that phenomenon called anticipation, right? Where the disease got worse or started earlier in each generation.
Exactly. And it just didn't fit Mendelian roles. The big breakthrough didn't come until 1991.
1991, what happened then?
That's when researchers finally identified the expanding trinucleotide repeat as the cause of Fragile X syndrome.
Ah, so that was the moment they realized the mutation wasn't fixed, it was dynamic, it could change.
Precisely. And since then, the floodgates opened. We've now linked about 40 different human diseases to these unstable repeats, and, that number is still climbing.
40 diseases. And is there a common thread?
Well, what's really striking, clinically speaking, is that almost all the trinucleotide repeat disorders discovered so far are associated with neurodegenerative changes, progressive damage to the nervous system.
Why the brain? Why is the nervous system seemingly the main target for these kinds of mutations?
That's a great question. Neurons are, well, they're particularly vulnerable. Most of them are postmitotic, meaning they don't divide and replace themselves after we're born. They have to last a lifetime.
Right.
So if they start accumulating toxic proteins, or if they lose a protein that's critical for their function, that damage builds up over years and years. It's cumulative and permanent. This long-term stress caused by the unstable DNA just seems to hit neurons harder than other cell types that can regenerate.
Okay, that makes sense. Before we dive into specific examples like Fragile X, we should probably nail down the general rules governing these disorders. You mentioned three key principles. The first one was the threshold effect.
Yes, absolutely fundamental. Whether or not someone gets the disease and how severe it is depends entirely on the number of repeats. It has to cross a certain threshold.
It's like a switch gets flipped.
Pretty much.
Yeah.
There's usually a normal range of repeats, perfectly stable, no disease. Then there's often a premutation range. Here, the repeats are unstable, they can expand, but they might not cause the full disease themselves, though they can cause other related problems we'll get into.
Okay.
And then finally, you cross into the full mutation range, and that's when you see the clear clinical symptoms of the disorder.
And we're talking about a difference that could be just a few repeats, right? I think in Huntington's, like 35 CAG repeats might be okay, but hit 39.
And the disease is essentially inevitable, though the age of onset can still vary. That threshold is critical. The second principle, parental proclivity, or you could call it a gender bias in expansion, is maybe even more peculiar.
This is where it matters whether you inherit the faulty gene from your mother or your father.
Correct. The tendency for that repeat sequence to suddenly jump, to really expand dramatically, often depends on the sex of the parent who passes on the premutation.
Can you give an example?
Sure. For Fragile X syndrome, the big expansion, say going from 100 repeats in a carrier mother up to 400 or more in her child. That amplification happens primarily during a telogenese.
So in the formation of the mother's egg cells.
Yes. The female reproductive process is where the major expansion occurs for Fragile X.
But for Huntington's, it's the other way around.
Exactly. For Huntington's, and actually for many of the polyglutamine disorders, the really large expansions are much more likely to happen during spermatogenesis.
In the father's sperm production, why the difference?
It's thought to be related to differences in how DNA is replicated and repaired during egg formation versus sperm formation. Sperm production involves many, many rapid cell divisions, perhaps giving less time for DNA repair mechanisms to fix those tricky secondary structures compared to the more prolonged, paused process of oogenesis.
Fascinating. Okay, the third principle you mentioned is about the actual mechanisms, how these repeats cause disease. You said there are 3 distinct ways.
Yes, and this is crucial because you can't treat all these disorders the same way. The location of the repeat dictates how it damages the cell. It creates cellular chaos in basically 3 different ways.
Okay, lay them out for us. What's the first category?
First is loss of function. This typically happens when the expanded repeat is in a non-coding part of the gene, maybe the promoter region that turns the gene on, or an intron, a section that gets spliced out.
So not in the part that actually codes for the protein.
Right. The huge expansion in that non-coding region leads to what's called transcriptional silencing. The cell basically shuts the gene down, often through a process called DNA methylation.
So the gene gets turned off.
Correct. And the disease is caused by the lack of the protein that gene was supposed to make. Classic examples are Fragile X syndrome and Friedreich ataxia.
Okay, loss of function. What's the second mechanism?
Second is toxic gain of function, but this time it's due to an altered protein. This is what happens in Huntington disease and the spinocerebellar ataxias. Here, the repeat expansion is in the coding region of the gene.
So it directly changes the protein blueprint.
Exactly. The gene gets transcribed into RNA and then translated into protein. But because the repeat, usually CAG in these cases, codes for the amino acid glutamine, you end up with a protein containing a long, sticky string of glutamines, a polyglutamine tract.
And that abnormal protein is toxic.
Yes, it misfolds, it clumps together, and it acquires some new harmful activity that poisons the cell. It might interfere with the normal version of the protein or just gain a completely novel toxic function.
Okay, loss of function, toxic protein. What's the third way? This one's newer, right?
Relatively newer, yes. It's also a toxic gain of function, but this time it's mediated by the RNA itself.
The messenger RNA? How can RNA be toxic?
In these cases, like the Fragile X premutation disorders or myotonic dystrophy, the repeat is again in a non-coding region, so the protein itself might be okay or even made at normal levels. But the messenger RNA molecule that's transcribed from the gene carries that expanded repeat sequence. And that expanded repeat within the RNA molecule makes the RNA itself sticky and problematic. It folds into structures that act like molecular flypaper or a sponge.
A sponge. What does it soak up?
It sequesters essential RNA-binding proteins. These are proteins the cell needs to process other RNAs, regulate splicing, transport mRNAs, basically manage the flow of genetic information.
So the toxic RNA traps these crucial regulatory proteins.
Precisely. It pulls them out of circulation, preventing them from doing their normal jobs. This widespread disruption of RNA processing is what causes the disease. Examples are Fragile X-associated tremor ataxia syndrome, or FXTAS, and myotonic dystrophy.
Wow. Okay, so summarizing, the gene gets silenced, the protein product poisons the cell, or the RNA intermediate messes everything up. 3 very different ways for a simple repeat expansion to cause havoc. ## polyglutamine diseases, coding region expansions. Let's dig a bit deeper into that second mechanism, the toxic protein gain of function. This seems to be behind many of the well-known late-onset neurodegenerative ones, like Huntington's. These are mostly the CAG repeats, right?
That's right. This whole group is often called the polyglutamine diseases because the CAG repeat codes for glutamine. When that polyglutamine tract in the protein gets too long, usually beyond 36 to 40 repeats, you see this consistent pattern across different diseases like Huntington's, caused by the HTT gene, various spinocerebellar ataxias or SEAs caused by different ATXN genes, spinobulbar muscular atrophy,
And the pattern is?
progressive neurodegeneration, typically starting in middle age.
Taking Huntington's as the classic example, the abnormal Huntington protein gets made. How exactly does it become toxic? What does it do inside the neuron?
It's complex and probably involves multiple hits. The long polyglutamine tract makes the protein prone to misfolding. Then it often gets cut up by cellular enzymes into smaller fragments.
And those fragments are the real problem.
Increasingly, evidence points to these smaller, soluble fragments as being particularly toxic. They don't just sit there, they actively interfere with vital cellular processes.
Like what?
Well, they're known to enter the nucleus and mess with transcription, the process of reading DNA to make RNA. They can bind to and inhibit crucial transcription factors, proteins that are needed to turn other essential genes on.
So the toxic protein shuts down the expression of other genes the neuron needs to survive.
Exactly. For instance, they interfere with factors like CBP and TFI-I130, which are critical for expressing genes involved in neuronal survival pathways. They also disrupt mitochondrial function, the cell's energy production, leading to an energy crisis. And they contribute to excitotoxicity, making neurons overly sensitive to damage from neurotransmitters. It's really a multi-pronged attack.
A cascade of failures leading to neuronal death, particularly in specific brain areas like the striatum and Huntington's.
Precisely.
Now, you often see pictures in textbooks. The hallmark finding under the microscope is these large clumps, these intranuclear inclusions of the mutant protein. It looks like the cell is trying to bundle up the trash.
That's the characteristic morphological feature, yes. These dense aggregates packed inside the nucleus of affected neurons. But their role is actually quite controversial.
Controversial? How so? I always assumed they were just bad.
Well, while their presence clearly marks the disease process, there's a serious debate about whether the inclusions themselves are the primary toxic species or if they might actually be protective.
Protective? How could a big clump of garbage protein be protective?
The hypothesis is that forming this large, relatively inert aggregate, the inclusion body, might be the cell's way of safely sequestering the truly toxic forms of the protein, which might be those smaller soluble fragments or misfolded monomers we just talked about.
So the inclusion is like a designated holding pen, trying to keep the dangerous stuff out of general circulation.
That's the idea. If the inclusion is a sort of tomb for the most toxic species, then trying to develop drugs that prevent aggregation, which was an early therapeutic strategy.
And actually make things worse.
It could potentially backfire, yes. If you stop the formation of these large aggregates, you might end up with higher levels of the more mobile, dangerous, soluble forms floating around in the nucleus and cytoplasm.
Wow. So researchers are trying to figure out whether to block clumping or encourage it. That sounds like a tricky line to walk.
It definitely demands caution. The focus is shifting a bit now towards enhancing the cell's natural clearance pathways, like autophagy, the cell's waste disposal system, to get rid of the misfolded protein, rather than just focusing on aggregation itself. We really need to pinpoint which form of the mutant protein is the main culprit before we can intervene effectively.
Okay, let's pivot now to the first mechanism you described, loss of function, using Fragile X syndrome as our main example. Why is FXS so important from a clinical standpoint?
It's incredibly significant due to its frequency. It's the most common inherited cause of intellectual disability in males. Overall, it's second only to Down syndrome, which is usually not inherited.
And the numbers?
Estimates vary, but it affects roughly 1 in 1,550 males, maybe 1 in 4,000 to 1 in 8,000 females, though female presentation is more variable. Because it's inherited, often through unaffected carriers, it has huge implications for genetic counseling and families.
The name itself, Fragile X, sounds quite evocative. Where did that come from?
It's a historical term, actually. It came from early cytogenetic studies, looking at chromosomes under a microscope. Researchers noticed that if you grew cells from affected individuals in a special culture medium, one deficient in folate, the X chromosome often showed a gap, or constriction, a point that looked like it was about to break off near the end of the long arm, at position XQ27.3. So they call it a fragile site.
But we don't diagnose it by looking for fragile sites anymore, do we?
No, not really. That method was tricky and not always reliable. Today, diagnosis is all molecular. We use DNA analysis like PCR or Southern blotting to directly count the number of CGG repeats in the FMR1 gene.
So you get a precise number.
Exactly. And that number tells you if it's normal, a pre-mutation, or a full mutation. It's much more accurate for diagnosis and understanding the risk.
Can you describe the typical features of the phenotype, especially in males who have the full mutation?
Well, the main feature is intellectual disability, which can range from mild to severe, but it's often quite significant. As they grow, especially after puberty, they tend to develop a characteristic appearance, often a long face, a prominent jaw, large ears that might stick out a bit.
Okay.
But the most distinctive physical sign, almost pathognomonic, is macroorchidism.
Meaning large testicles.
Yes, significantly larger than average. This is seen in at least 90% of affected males after puberty. It's a key diagnostic clue.
And neurologically, it's more than just intellectual disability, right? There are often other issues.
Oh yes, the comorbidity is high. About 30% of individuals with FXS experience epilepsy. Behavioral issues like hyperactivity, anxiety, and sometimes aggression are very common, reported in maybe up to 90%.
And there's a strong link to autism.
A very strong link. FXS is the most common known single-gene cause of autism spectrum disorder. Estimates suggest that maybe 2 to 5% of all individuals diagnosed with autism, and especially those without other clear syndromes, actually have the FMR1 mutation underlying it. Hashtag the FXS inheritance paradox. Anticipation in carriers.
Now let's circle back to that confusing inheritance pattern you mentioned earlier, the anticipation effect, and how the discovery of the triplet repeat finally cracked the code.
Right. Before 1991, FXS inheritance was a real puzzle. It didn't follow the rules for a typical X-linked recessive disorder where you'd expect carrier females to be mostly unaffected and affected males to inherit it from carrier mothers.
What was weird about it?
Two main things. First, there were these normal transmitting males. These were grandfathers who carried the genetic defect but were completely unaffected themselves, clinically normal.
Okay.
But they would pass the faulty gene to their daughters, who were also usually unaffected carriers. But then the sons of those daughters, the grandsons of the original transmitting male, were often severely affected with FXS.
So it looked like it skipped a generation, but only when passed through a female.
Exactly. That didn't fit. Second, a surprisingly high percentage of the female carriers were affected to some degree. About 30% to 50% of females carrying the full mutation show some level of intellectual disability or learning problems.
Which is much higher than you'd expect for a standard X-linked recessive condition where females are usually just carriers.
Right. Because females have 2 X chromosomes, they usually have one normal copy of the gene to compensate. But in FXS, that compensation often isn't enough, or maybe X inactivation patterns play a role.
So how did the triplet repeat explain all this? Let's revisit those size ranges again.
Okay, so the normal range is small and stable, about 6 to 55 CGG repeats in the FMR1 gene. No problems here.
Got it.
The premutation range is bigger, roughly 55 to 200 repeats. This is what the normal transmitting males and the carrier daughters typically have. The gene is still mostly functional, but this repeat size is unstable. It can expand.
And then the full mutation.
That's when it crosses the threshold, usually considered to be over 200 repeats, and can go to 4,000 or even more. This is the range that causes Fragile X syndrome.
So the normal transmitting male, the grandfather, has a premutation, maybe 80 repeats. He passes it to his daughter. She also has 80 repeats, still a carrier. But then
When she forms her egg cells during eugenesis, that premutation becomes highly unstable. There's a significant risk that the 80 repeats will massively expand, jumping up to, say, 500 repeats in the egg cell.
wow.
If that egg is fertilized, especially if the resulting child is male, XY, he inherits that X chromosome with the huge expansion, the full mutation, and develops FXS.
So the expansion happens primarily when the mother passes the gene on?
Yes, the probability of a premutation expanding to a full mutation is much, much higher during eugenesis than during spermatogenesis. That's why the grandsons were affected, but the sons of the transmitting males weren't. This dynamic mutation, the expansion during maternal transmission, was the key that unlocked the whole mystery of anticipation and the weird inheritance pattern of FXS. Okay, we've established that the full mutation, over 200 repeats, causes FXS through loss of function. How exactly does that huge number of CGG repeats physically turn the FMR1 gene off?
It's an epigenetic mechanism. When the number of CGG repeats gets very large, usually crossing a threshold around 200, 230 repeats, it triggers a massive chemical modification of the DNA in that region.
What kind of modification?
Abnormal DNA methylation. The cell starts adding methyl groups, little chemical tags, onto the cytosine bases, the C in CGG, within the repeat sequence itself, and also onto the promoter region of the FMR1 gene, the part that normally acts like the on switch.
And methylation is like a silent signal for genes, isn't it?
It's one of the cell's primary ways to permanently shut down gene expression. This dense methylation effectively locks the gene in an off state.
Resulting in?
Transcriptional suppression. The cell can no longer read the FMR1 gene to make messenger RNA, and if there's no mRNA, there's no protein. So individuals with the full mutation have little or no fragile X mental retardation protein, or FMRP. FMRP. So the absence of this one protein is the direct cause of all the symptoms of Fragile X.
Largely, yes. The loss of FMRP function is the root cause of the intellectual disability and other features.
So what does FMRP normally do? Why is it so important, especially in the brain?
FMRP is actually quite abundant in the brain and also in the testes, which might explain the macroorchidism feature. In neurons, its main job is incredibly sophisticated. It acts as a translation regulator.
Translation regulator, meaning it controls protein production.
Exactly. Specifically, it controls the local production of proteins right near the synapses, the junctions between neurons. This local control is critical for learning and memory, for synaptic plasticity.
How does it work?
FMRP binds to a specific set of messenger RNAs, hundreds of them, actually that code for proteins involved in building and modifying synapses. It acts like a chaperone, escorting these mRNAs out from the cell body along the dendrites to the sites where synapses are formed.
So it delivers the instructions for building synaptic parts right to the construction site.
Precisely. But it does more than just deliver. Once there, FMRP typically acts as a brake, actively suppressing the translation of those mRNAs into protein.
It stops the protein from being made. Why?
It keeps protein production on hold until the synapse receives a specific signal, a trigger. One key trigger seems to be activation of certain receptors on the neuron surface, specifically group I metabotropic glutamate receptors, or mGALIRs. When these receptors are activated, the brake is released, and the needed synaptic proteins are synthesized on demand.
Okay, so FMRP is delivering the instructions but keeping them paused until the go signal arrives. What happens in Fragile X when FMRP is missing? The brake is gone.
The brake is gone. So you get excessive, uncontrolled translation of all those synaptic protein mRNAs that FMRP normally keeps in check.
Too much protein synthesis at the synapse.
Yes, and this leads to observable abnormalities in the structure of synapses. Instead of the mature mushroom-shaped dendritic spines you see in typical development, neurons in FXS tend to have an overabundance of long, thin, immature-looking spines.
So the connections are physically different, less mature.
Exactly. They're thought to be less stable and less capable of undergoing the changes needed for learning and memory, that process called synaptic plasticity. This underlying defect in regulating synaptic protein synthesis and the resulting abnormal synaptic structure and function is believed to be the core reason for the intellectual disability, the learning problems, and maybe even some of the autistic features seen in FXS.
And that understanding actually points towards potential treatments, doesn't it? Targeting that signaling pathway.
It does. A lot of research is focused on using drugs that block or dampen the activity of those mGALIR receptors. The idea being to reduce the excessive signaling and bring that runaway protein synthesis back under control. Hashtag tag the paradox of the premutation. Toxic RNA gain of function.
But the story gets even more complex when we look back at the carriers, the people with the premutation 55 to 200 repeats. They don't have Fragile X syndrome itself, but they're not necessarily unaffected, are they?
Not at all. This is one of the most fascinating and frankly paradoxical aspects of the FMR1 gene. Individuals with the premutation are at risk for developing two completely distinct clinical disorders, which manifest much later in life and are caused by that third mechanism we talked about, toxic RNA gain of function.
Okay, so different mechanism, different diseases. What are they?
First, primarily in male carriers, there's FXTS, which stands for Fragile X-Associated Tremor Ataxia Syndrome.
Tremor ataxia.
Yeah.
Sounds neurological.
It is. It typically starts after age 50, maybe affecting up to 50% of older male carriers. It's a progressive neurodegenerative condition causing intention tremor shaking that gets worse with purposeful movement and cerebellar ataxia, which means problems with balance and coordination.
And the second disorder affects female carriers.
Yes, that's FXPOI, or Fragile X-Associated Primary Ovarian Insufficiency. This affects about 20% of female premutation carriers.
And that means?
It causes their ovaries to fail much earlier than normal, leading to infertility and premature menopause, often before the age of 40.
So let me get this straight. The same gene, FMR1, depending only on the number of repeats, can cause severe intellectual disability from birth, FXS, full mutation, or late-onset neurological problems and ovarian failure, FXDASS, FXPOI, premutation. How can the mechanism be so drastically different?
It all comes back to that key difference, methylation. In the full mutation, 200 repeats, the gene gets heavily methylated and silenced. No FMRP is made. Loss of function.
Right.
But in the pre-mutation range, 55, 200 repeats, that massive methylation doesn't happen. The FMR1 gene remains active. It is transcribed into messenger RNA. In fact, it's often transcribed at even higher levels than normal.
More RNA is made. Why isn't that good?
Because the RNA itself is the problem. This pre-mutation mRNA contains that expanded stretch of CGG repeats, and that expanded CGG sequence within the RNA molecule makes the RNA adopt unusual, stable secondary structures, like hairpins.
Okay.
These structures turn the mRNA into a toxic entity. They act like a trap or that molecular sponge we mentioned earlier.
Sequestering those RNA-binding proteins again.
Exactly. The toxic CGG repeat containing mRNA binds very tightly to various essential RNA-binding proteins, pulling them away from their normal jobs in the cell. These proteins get stuck to the toxic RNA, sometimes forming visible clumps or intranuclear inclusions within the cell nucleus, a hallmark of FXTS pathology.
So the cell is being starved of these regulatory proteins because they're all tangled up with the faulty FMR1 mRNA.
That's the essence of it. Proteins crucial for things like splicing, cutting and pasting other RNA molecules correctly, RNA transport, and general gene regulation get sequestered. This widespread disruption of RNA metabolism is what's thought to cause the neuronal dysfunction and death, leading to FXTS symptoms, and also the dysfunction in the ovary, leading to FXPOI.
So it's a complete flip. In FXS, full mutation, the problem is too little FMRP protein. And at FXTS, FXPOI permutation, the problem is too much toxic FMR1 RNA.
Precisely. A beautiful, if devastating, example of how the quantity of a genetic repeat can fundamentally change the quality of the disease mechanism. #MishTA. Summary of other trinucleotide disorders in conclusion.
This framework, loss of function, toxic protein, toxic RNA, is clearly essential for making sense of this whole group. We've gone deep on Fragile X and touched on Huntington's. Maybe we should briefly highlight a few other key examples to really cement these different mechanisms.
Yes, looking at other disorders helps illustrate the variations, like different repeat sequences or slightly different twists on the mechanisms.
Okay, how about another example of toxic RNA gain of function, but one that doesn't involve the FMR1 gene? Myotonic dystrophy comes to mind.
Good one. Myotonic dystrophy type 1, or DM1, is actually the most common form of muscular dystrophy that starts in adulthood. It's a multi-system disorder affecting skeletal muscle, heart muscle, the brain, eyes, endocrine system.
And the repeat isn't CAG or CGG here, is it?
No, in DM1, the culprit is an expanded CTG repeat. And importantly, it's located in the 3' untranslated region, 3' UTR, of the DMPK gene, another non-coding region.
Okay, non-coding region suggests either silencing or toxic RNA. What's the repeat count threshold?
Normal is about 5 to 37 CTG repeats. Disease can start with maybe 50 to 100, but severe congenital forms can have thousands of repeats.
In the mechanism, is it like FXTS?
It's very analogous, yes.
Yeah.
Primarily toxic RNA gain of function. The DMPK gene gets transcribed, producing mRNA containing a long CUG repeat tract, because tNDNA becomes U in RNA.
CUG repeats in the RNA.
And these expanded CUG repeats in the mRNA act as that molecular sponge again.
Mm-hmm.
This time they specifically sequester a family of proteins called muscle-blind-like proteins, or MBNL proteins.
MBNL proteins, what do they normally do?
They are crucial regulators of alternative splicing, especially muscle and nerve cells. Splicing is how the cell cuts and pastes bits of RNA to create different protein versions from the same gene.
So the toxic CUG RNA traps the MBNL splicing factors,
preventing them from doing their job on hundreds of other target RNA throughout the cell. This leads to widespread missplicing. Many genes start getting spliced incorrectly, often reverting to patterns normally only seen in fetal development.
Wow, systemic splicing chaos.
Exactly. And this missplicing of key proteins like the chloride channel, causing the myotonia-delayed muscle relaxation,
Right.
the insulin receptor, contributing to diabetes risk, and cardiac proteins, causing heart conduction problems, explains the diverse symptoms of DM1. It's fundamentally a disease of faulty RNA processing caused by the toxic repeat RNA.
Okay, that's a powerful example of toxic RNA. Now let's switch gears to a pure loss of function example that doesn't use CGG repeats like Fragile X does. How about Friedreich ataxia?
Perfect example. Friedreich ataxia, or FRDA, is the most common inherited ataxia diseases affecting coordination. It causes progressive damage to the nervous system, particularly the spinal cord tracts involved in movement and sensation, and also significantly affects the heart.
And the repeat here is different again.
Yes, FRDA is caused by an expanded GAA repeat, and this repeat is located in the first intron again, a non-coding region of the FXN gene.
Intron, GAA repeats. What are the numbers?
Normal individuals have about 7 to 34 GAA repeats. The disease occurs when the expansion goes over, say, 65 repeats, but it often involves hundreds, sometimes over 1,000 repeats.
So non-coding region, massive expansion. Sound like loss of function, similar to Fragile X mechanism. Is it methylation again?
It's loss of function, but the mechanism of silencing seems a bit different from Fragile X. While some epigenetic changes might occur, the primary problem with the huge GAA repeat tract seems to be structural. It causes the DNA itself to adopt an unusual, very stable triple helix structure.
A DNA triple helix instead of a double helix.
Yes, or other non-standard structures, often called sticky DNA. This abnormal structure acts as a physical roadblock, directly impeding the RNA polymerase enzyme from transcribing the gene.
So it just blocks the gene from being read.
Effectively, yes. It severely reduces the amount of FXN mRNA that gets made,
Resulting in too little of the protein. What protein is it?
The FXN gene codes for a protein called frataxin.
Frataxin. What's its job?
Frataxin is vital for mitochondria, the energy factories of the cell. Specifically, it's involved in building iron-sulfur clusters, which are essential components of the mitochondrial machinery involved in energy production and handling iron.
So low frataxin means problems with mitochondria and iron.
Exactly. The lack of frataxin leads to iron overload inside the mitochondria, which generates massive oxidative stress, damages mitochondrial function, and ultimately leads to cell death, particularly in high-energy demand tissues.
Like neurons in the spinal cord and heart muscle cells.
Precisely why you see the characteristic ataxia and cardiomyopathy in FRDA. It's a loss-of-function disease caused by reduced frataxin due to transcriptional blockage by the GAA repeat structure.
Okay, so looking across these examples, Huntington's toxic protein, Fragile X, loss of function via silencing, myotonic dystrophy, toxic RNA via sequestration, Friedreich ataxia, loss of function via transcription blockage. What's the big takeaway?
The unifying feature is clearly the unstable DNA repeat. But the crucial point is that how that repeat causes disease is fundamentally different depending on the specific repeat sequence, CAG, CGG, CTG, GA, etc. And critically, its location within the gene, coding versus non-coding, promoter versus intron versus UTR.
Silencing the gene, making a toxic protein, or making toxic RNA.
Right. And understanding which of those three paths a particular disease takes is absolutely essential if we ever hope to develop effective therapies.
It seems incredibly complex, but also, in a way, elegantly distinct. Why should someone listening, maybe not a geneticist, really care about distinguishing between, say, a toxic protein mechanism and a toxic RNA mechanism?
You should care because it dictates the entire strategy for designing a treatment.
Hmm.
If you have a loss-of-function disease like Friedreich ataxia, maybe you try to develop drugs that boost the transcription of the FXN gene or perhaps deliver functional protaxin protein. Gene therapy might be an option. But if you have a toxic RNA disease like myotonic dystrophy, your target is completely different. You might design small molecules or antisense oligonucleotides, custom-made bits of DNA or RNA that specifically bind to and neutralize that toxic CUG repeat RNA, freeing up the sequestered MB and L proteins.
While leaving the DMPK gene itself alone.
Ideally, yes.
Yeah.
Or for a toxic protein disease like Huntington's, you might try to reduce the production of the mutant Huntington protein using gene silencing approaches, or enhance its clearance, or maybe try to block its downstream toxic effects. The therapeutic approach has to match the underlying molecular pathology.
Get the mechanism wrong and the therapy is doomed from the start.
Absolutely. Understanding the mechanism is the critical first step towards rational drug design and finding a cure, moving beyond just managing symptoms.
It really is remarkable, thinking back to Fragile X, that the difference between having, say, 150 repeats causing a late-onset tremor via toxic RNA, FXES, and 250 repeats causing profound intellectual disability from birth via gene silencing, FXS, is just that threshold crossing. Same gene, radically different outcomes based on repeat number dictating the mechanism.
It highlights the incredible sensitivity of these biological systems. And that brings us perhaps to a final thought for you, the listener, to consider.
Go on.
Given that we can now pinpoint these different mechanisms, toxic protein, toxic RNA, gene silencing, and we understand they stem from the same fundamental problem, the unstable repeat. How close are we really to developing therapies that target the root cause? Could we, for instance, use technologies like CRISPR gene editing, not to fix the downstream consequences, but to actually go into the DNA and precisely snip out or shrink that unstable repeat before it expands past the toxic threshold?
treating the genetic instability itself rather than the diverse fallout.
Exactly. Could we prevent these diseases entirely by correcting the DNA typo before it causes harm? The potential is enormous, but so are the challenges of doing it safely and effectively in humans. The power, though, lies locked within understanding the dynamics of just those few hundred repeating letters of DNA.
A truly provocative thought. It's precision medicine taken to the ultimate level, preventing the disease before it even starts. Thank you. This has been an incredibly insightful journey through a really complex area of genetics.
My pleasure. It just shows that beneath the complexity, there are often elegant molecular rules, and understanding those rules is the key to making progress.